Hello, today we're going to go over chapter 22, the lymphatic system, from Fundamentals of Anatomy and Physiology by Frederic Martini. So what are our objectives for today? We're going to look at the structure of the lymphatic system. We're going to talk about non-specific and specific components of the lymphatic system. Whenever we talk about the specific components, we're going to talk about cell-mediated and antibody-mediated immunity. We're going to look at specifically T cells and B cells, and we're going to finish up the chapter looking at the role of stress and the relationship between stress, aging, and your lymphatic system, which is your immune system, which is something that in the age of that is why certain particular demographics can be susceptible to specific diseases such as COVID-19. Okay, so the lymphatic system contains, we're going to talk about the cells, tissues, and organs of the lymphatic system. Their role is to defend us against foreign invaders. What could be considered foreign invaders? You have your bacteria, you have viruses, you also have cancer cells, and you have substances released by bacteria, such as the cholera toxin. Here is an overview of the structure of the lymphatic system. It contains organs of the lymphatic systems as well as the vessels. Here, we have the spleen, you have tonsils, you have your thymus. Those are part of the organs of the lymphatic system. You also have your mucosa-associated lymph tissue as part of these organs of the lymphatic system. You also have these vast lymphatic vessels that are specific, that have lymph nodes specifically located in areas of high convergence or areas that are exposed to the exterior environment. So areas that are exposed to the exterior environment would be your digestive, respiratory system, and areas of high convergence would be your axillary node surveying your arms, your cervical node surveying your head and neck, your inguinal node surveying your lower extremities. So let's look at the hierarchy of the structural components of the lymphatic system. You have your lymphocytes and supporting phagocytic cells. So these are cells that engulf foreign invaders, but the role doesn't stop there. Whenever cells engulf and the foreign invaders, they don't just digest it and kill it. They also save portions of it, and they display them on their membrane and say, "Hey, we're under attack by this guy." So basically, these macrophages they help activate the immune system. They help present the foreign invader in their surface and say, "Hey, this is the attacker. We need to boost our response specifically against this. I ate this, and this is how he looks like. This is how the foreign invader looks like." All right. You also have, I got a little carried away. You also have the lymph, which is where these lymphocytes are going to be exposed to foreign invaders. We're going to look at the relationship between blood, interstitial space, and the lymphatic system, and how this sequence allows the lymph vessels, the lymph itself, to survey of our body for foreign invaders. You have the lymphatic vessels carrying the lymph, and you have your lymphatic tissues and organs. The organs can be divided in primary and secondary lymphoid organs. Your primary lymphoid organs are going to be places where you have your lymphoid stem cells dividing and giving rise to B lymphocytes in the bone marrow and T lymphocytes in the thymus. You also have your secondary lymphoid organs, where there are these are sites of accumulation of lymphocytes, and they're performing this survey role. You have your lymph nodes, your tonsils, spleens, and your mucosa-associated lymphatic tissues present in mucosas. Mucosas that are present there to provide mucus to surfaces. Surfaces which within our bodies, they're usually lumens. They're usually tubes, right? Tubes of the respiratory system, tubes of the digestive system. Okay. The function of the lymphatic system, the function is we have that immune function. This immune function, they not only have to detect the foreign invader, but they have to be able to reach it. So a foreign invader can be anywhere in our body. So the lymphatic system has to exist in all places in our body. Usually, the lymphatic vessels and capillaries are associated with our vasculature, our arteries and veins. And arteries and veins, they reach everywhere because cells, they need oxygen. They need it all the time, and they need nutrients. They need nutrients as well. So blood vessels are always near a cell, and capillaries are always going to be near cells to perform their roles. Whenever we talk about vessels of the lymphatic system, you have your lymphatic capillaries, you have your small lymphatic vessels, and you have these major collecting vessels that deposit into trunks. Okay. So here are your lymphatic capillaries. This shows the interplay between circulation, the circulatory system, your vascular system. So basically, the vascular system tries to provide oxygen, nutrients, ions, the right environment for cells to have what they need in terms of gases, nutrients, and the right environment to thrive for homeostasis. This exchange occurs due to differences in pressure. Now, those same differences in pressures don't permit everything that went out due to differences in pressures to return into the vascular system. So some of the liquid is the same due to difference in pressure. There's lower pressure in the interstitial, there's lower pressure in the lymphatic fluid than the interstitial space. So just naturally, you're gonna have fluid and foreign invaders, and in some cases lipids go into the lymphatic system. And this is the lymphatic system is analogous to veins. The lymphatic vessels are analogous to veins in terms that they have valves within them. And so here you have this is how lymphatic capillaries looks. They have this epithelium present here that is lined up like tiles of a roof in a roof. So whenever there's enough pressure here, the fluid will flow in between these tiles. Here is an example of valves. So here in this example, you will have fluid flowing in this direction from left to right of the screen, your right. And this valve here is preventing backflow. So whenever it comes to the drainage of the lymphatic system, I want to talk about these two major draining systems. You're going to have your thoracic duct and your right lymphatic duct. The right lymphatic duct is going to receive input from the right arm, right portion of the thorax, and the right portion of the head. The thoracic duct is going to receive input from the rest of the body. So just knowing this chart, you will know what trunks drain where. So in this case, we talk about the right lymphatic duct. It receives drainage from the right arm through the right subclavian trunk, drainage from the right thorax, from the right bronchiomediastinal trunk, drainage from the right head and neck from the right jugular trunk. It's a mirror trunks happen on the other side, draining into the thoracic duct. We also have the intestinal trunk draining the digestive system, and we have the right and left lumbar trunk draining the lower extremities. Here is a view of the trunks of the different trunks. So here's the left lumbar trunk, right lumbar trunk, the intestinal trunk, draining into the cisterna chyli, which is contiguous with the thoracic duct. The thoracic duct itself eventually drains into the subclavian vein. So all of that fluid that was collected in the lymph after it goes through the lymphatic system, all of those lymph nodes with lymphocytes surveying the system, you have it return to the cardiovascular system at the level of the subclavian vein. You see it happening here, and the right lymphatic duct also does the same. Here's a view of the cisterna chyli and the thoracic duct. We talked about the vessels. Now let's talk about the cells of the lymphatic system, the lymphocytes. There are 3 types: T, B, and natural killer cells. So you have your T cells. They do hand-to-hand combat. And you have your B cells. They are the ones that provide that humoral immunity, that humoral meaning that fluid, immunity within fluid. And this one is antibody mediated. So you have B cells differentiated into plasma cells, which can produce antibodies. And you also have natural killer cells. These are cells that they kill cells within our bodies. They kill our own cells whenever they are sick. So if a cell is infected by a virus, or if a cell shows cancerous epitopes, cancerous proteins within its surface, natural killer cells are that part of our immune system, are lymphocytes that will kill this. So these are the different types of T cells. And we talked about B cells and plasma cells. We're going to talk about this later whenever we talk about T cells, but I want you to become familiar with some of these names. Your cytotoxic T cells, your helper T cells, your suppressor T cells, and your memory T cells. Cytotoxic T cells, these are the ones that are responsible for this hand-to-hand combat that we talked about. Your helper T cells, they are responsible for summoning the immune system. These are saying, help, we found these. B cells, help us. T cells, help us. So they're the ones that are basically mediating and also making sure that we are attacking the right foreign invader. Suppressor T cells, they suppress the immune system. This natural upregulation of immune cells will be downregulated eventually. Suppressor T cells help with this. And memory T cells, whenever we find a foreign invader, we attack it, we get rid of it, we keep a copy of it. So next time that flu returns, or if we're vaccinated and we have that initial response, the next time we have a memory cell ready to fight it again. Okay, so these are what we're seeing here is we're seeing basically the creation of these lymphocytes. We talk about B lymphocytes being developed in the bone marrow, and T lymphocytes being the lymphoid stem cells residing in the thymus. So you have this kind of multipotent stem cells. These stem cells that can give rise to multiple stem cells or multiple cell types. This differentiates into a lymphoid stem cell that gives rise to B cells within the bone marrow, and a lymphoid stem cell that travels to the thymus, and within the thymus it gives rise to T cells, and eventually these natural killer cells, B cells, and these T cells, they perform their function. You have here your natural killer cells performing immune surveillance. So whenever it finds a cell that's being attacked by a virus, or it's cancerous, it's surveilling our body, and whenever it finds these cells, it attacks them. You have your B cells maturing into plasma cells and releasing antibodies, and you have your T cells, which attack cells cells or foreign invaders. It can attack bacteria. It can attack other components just in a cell-to-cell manner. So we talked about the vessels. We talked about the cells. Now let's talk about the lymphoid tissues. So lymphoid tissues are basically connective tissues that are rich in lymphocytes. So this happens in areas that are exposed to foreign, where there's a greater likelihood of foreign invader exposure. This is here you have an example of mucosa. This happens on mucosas, mucosa-associated lymphoid tissues. You're gonna find this on your respiratory system, your digestive system, portions on your reproductive system as well. Portions that are basically open to a lumen, where there might be high level of contact with foreign substances, invaders, and basically these mucosas, these mucosal layers, they're rich on these lymphocytes. You also have lymphoid nodules in areolar tissues. Here's an example of this would be your tonsils, your pharyngeal, your pharyngeal, palatine, and lingual tonsils. You have your palatine tonsils usually being the ones kind of at the forefront of contact with the exterior environment, so they're the ones that are most likely going to be infected, and most likely whenever it comes to removal, they're the ones that go out first. That also happens to your adenoid tonsils or your pharyngeal tonsils, and in rare cases to your lingual tonsils as well. Let's talk about lymphoid organs. You have your lymph nodes, your thymus, your spleen. The function of your lymph nodes is these are those check stations within your lymph. where you have high contact between lymphocytes and the lymph. So afferent lymphatics bring lymph into the nodes, and efferent lymphatics carry lymph from the nodes. We're going to look at this in depth. So this is a lymph node. You have your lymphocytes, your T and B lymphocytes located in specific regions within the lymph node. So here in the cortex, you see these follicles present here that contain B lymphocytes on the exterior, and they contain a germinal center in the middle that gives rise to more B lymphocytes. Now, these B lymphocytes are also present in these medullary cords. You also you will find T lymphocytes within this outer portion of the medulla. So in this dotted area, you will have T lymphocytes. Okay, so that is where the lymphocytes are present. I want you to keep that in mind. Now let's talk about just structure. You have here a capsule that infolds into the lymph node through this trabecula. Between the capsule and the cellular component here, there's a space. It's called the subcapsular sinus. So sinus being these spaces. This is through these sinuses, you have the lymph percolating and being exposed to these different lymphocytes. And the lymphocytes, if they have a receptor on their surface that is able to recognize this foreign invader, they're gonna go, I've seen this before. This is a pathogen. We need to attack it. So this is where this high level of contact between pathogens and the immune system occurs. And here you have the subcapsular sinus. This is a view of it. So this is the capsule. The afferent vessels bring all the lymph, and the lymph just percolates. It just washes over these sinuses, and the sinuses get exposed to the immune system in this manner. The dendritic cells present here, you're gonna hear a lot of names for macrophages or antigen presenting cells. So basically these T lymphocytes, in order for these lymphocytes, in order for them to recognize a foreign invader, they need a cell to show it to them. They need a cell that has digested that and put it on its surface. They need to be shown. Like that is a way for the immune system to make sure that we're attacking the right. It's like a way of checking and balancing that we're attacking the right substance. So these antigen presenting cells are the ones that are going to show the immune system what they need to attack. So these are going to be macrophages. They're going to be macrophages that have really specific names, such as microglia in the nervous system, dendritic cells within these lymph nodes, Kupffer cells within the liver, Langerhans cells within your epithelium. Those are all cells that digest something and put it on its surface and tell the immune system, tell the lymphocytes, I found this, attack it. So those are all, let's talk about them all as antigen presenting cells just for the sake of sanity and simplicity. Okay, so what do we have here? We have the afferent vessels bringing lymph, and the efferent vessels, after it percolates throughout all of these sinuses, it percolates, it gets exposed to the immune system, and the lymph eventually leaves through these efferent vessels. This region right here where blood vessels rush in and efferents live is called the hilus. All right, let's talk about another organ. Let's talk about the thymus. We talk about the thymus as being the site where the lymphoid stem cell for T cells or T lymphocytes resides. Okay, this is going to be found within the mediastinum. It's going to be behind the sternum and anterior to the heart and the trachea and major blood vessels. This is the structure of the thymus. It has two different lobes, and those lobes they're going to be separated into through these infoldings of the capsule is going to be divided into these lobules through these infoldings that are known as septa. So this is it secretes hormones that are responsible for the maturation of the T lymphocytes. So T lymphocytes, we talk about, you're going to see, for the most part, a theme within lymphoid structures. You have the core of these follicles, of these areas rich in lymphocytes, the core of it having a germinal center where there's the cell division when needed, and you have the outer portion having the functional lymphocytes. On the thymus is the other way around. You have on the exterior portion, you have the in this case the lymphoid stem cells that give rise to these T lymphocytes, and in the center of it you have your T lymphocytes present. Within this process of maturation, there is the presence of a blood. There is the presence of these thymic epithelial cells. Thymic epithelial cells that create a blood thymus barrier. You might have heard of a blood thymus barrier. Your fetal mother barrier, your blood-brain barrier. You also have a barrier within your spermatocytes and the blood. So basically, all of these barriers are trying to keep the blood away from a cell type that might be attacked, or it be your fetus, being an haploid sperm, or being in this case T cells that are not ready for to perform their immune function, that might be overzealous, and if exposed to blood, they might say, hey, this is foreign. So this barrier, how do this barrier, the way these barriers work is basically there are cells that just completely wrap around blood vessels. So they create they wrap around blood vessels and don't allow substances from the blood vessels to freely migrate into this particular tissue. So they create this barrier. The purpose of this barrier in this case is for the lymphocytes, the T lymphocytes, to completely mature. And this process of maturation involves making sure that they recognize our own cells and they don't attack them. 98% of lymphocytes that are undergoing this process of selection die by apoptosis because of this process. If they attack our own cells, there is a selection process that allow those lymphocytes to die. So this Within this is occurring, this barrier occurs here at the cortex, and within the medulla, where you have the functional, the ones that made it through this process within the medulla, you have these functional T cells. There are these components within the thymus called Hassel's corpuscles, and they're going to be just thymic epithelial cells creating these layers, and we don't know what they do. In general, nobody knows what they do. Okay, for certain. There's probably theories out there. So let's look at the spleen. The spleen, you're gonna have two components. You're gonna have a red pulp and a white pulp. The white pulp contains your components of the immune system, and your red pulp contains just components of the cardiovascular system. It's highly sinusoidal. Sinuses are these spaces, and the function of the spleen is the removal of abnormal blood, storage of iron. So we talked in the last chapter how blood gets recycled. Part of this recycling involves the recycling of this metal, iron. This happens at the spleen, and you have also the initiation of there's due to the large exposure of blood and these components of the immune system, you have activation of the immune system um, being activated by the circulating blood. We also have activation of the immune system whenever we're talking about the lymph nodes, but that's activated by the lymph itself. Here's a view of red pulp. Here's a view of white pulp. White pulp, there's usually a splenic artery at the center of, there's an artery at the center of these splenic nodules. So here you have the vascularization provided by the splenic artery. All of these infoldings of all of these marks in the spleen are going to be from its boundaries, its surroundings. All right, let's start talking about the specifics of the immune system. I know it's gonna go for a while. It's a lot. You can pause it here. This is a good way, this is a good place to take a little break. I'm gonna continue, but I encourage you to take a break, just digest it, breathe it in. We saw this like macro level of the immune system. It's good. Now we're going to transition gears and looking at the specific portions of it. We're gonna talk about specific molecules. We're gonna talk about pathways. So we're shifting gears at this moment, okay? Okay. Welcome back from your break. Now we're going to look at the non-specific and specific defenses of the immune system. The non-specific defenses basically are these defenses that apply to wide categories of foreign invaders, that apply to all viruses or all bacteria. The specific defenses, they're specific. They apply to not one virus, not one bacteria. They apply to a protein within that virus, a protein within that bacteria. This we'll later know them as epitopes. Okay, so part of the non-specific response include physical barriers. So these are your skin, epithelium within your digestive system. We have glandular secretions that allows us to maintain this, to start this first line of defense. We have lysozymes. We have the mucus itself, sometimes with the immune components within it, or simply the mucus itself creates a barrier that prevents direct contact. We have phagocytes, cells that just they just digest foreign invaders. We talked about last week about neutrophils, eosinophils, macrophages. These are all white blood cells that have this phagocytic component, and once they digest it, they usually present it. We have other categories of these macrophages known as fixed macrophages and free macrophages. I believe that we gave examples of fixed macrophages, microglia, Kupffer cells, and Langerhans cells for the nervous, liver, and skin, respectively. And you have free macrophages that are just freely circulating and just digesting foreign substances. And these macrophages, once they do, they usually present it to the immune system saying, I ate this, it didn't taste good, it's attacking us. Let's attack it. Let's get rid of it. So it summons your lymphocytes, your T, B lymphocytes. It summons the cavalry. We also, non-specific defense involves immunological surveillance, your natural killer cells. Basically, if there is a cell that is doesn't look healthy, either if it has some viral epitopes on its surface or some cancerous epitopes, cancerous antigens on its surface, and it will be attacked and destroyed by the natural killer cells. Here is the process of how the natural killer cells perform this function. This is a view of it. You have it recognizes, it adheres to the cell. Usually, this cell is sending a signal telling the natural killer cell, hey, I'm cancerous, or I have a virus that's attacking me. Sometimes the signal is specific, other times it just merely the cell doesn't have the right receptors on its surface because it's sick. So that's the part where it's not specific. So anyway, we recognize it adheres, then it aligns the Golgi apparatus in order for the Golgi to secrete these vessels. Whenever these vesicles will contain perforins. These perforins could create these complexes that just poke pores within the surface of the cell. We mentioned that cells are zealous about their homeostasis. They need a homeostasis to survive. And poking a hole on its surface is just an egregious breach of homeostasis. The cell will die. Non-specific differences. We have interferons. These are cytokines that basically are released and they have antiviral activities. So some of their antiviral activities involve it attracts natural killer cells, these cells that kill sex cells. It increases inflammation, which means it increases vascularization, so the likelihood of other components of the immune system it's summoning vasculature, and with it summoning components of the immune system. And you also have gamma interferon, which stimulates the activity of macrophages. So interferon, I want you to know that it has antiviral properties. Let's just do that. Okay, the complement system. So this is it's complicated. The immune system is just so complicated. But that's what keeps us from attacking ourselves, and it keeps us from basically just through random expression of genes of identifying foreign substances as foreign. It's amazing that we can identify something as foreign and not identify things that we need to survive as foreign. Like I know sometimes that happens, that we have these autoimmune responses and so forth, but for the most part, it works. But it's all about these lock and key type of mechanisms that allow this function to occur. So let's talk about the complement system. So basically, the target, the goal of it is to destroy the cell membranes, and it also has stimulates inflammation, attracts phagocytes, and enhances phagocytosis. It's made by this cascade of about 30 proteins, and we're going to see two pathways where how this occurs: the classical and the alternative. The classical is antibody triggered. So if there's an antibody on the surface of this particular component, uh, telling, just summoning the complement system, telling, "Hey, I found this. This is not part of our body. We need to attack it." So it activates the complement system in order for it to poke holes on and have those other effects into the cell. So here you have the complement system, just all of these 30 cascade, the 30 protein cascades that we mentioned. Oh, it attaches to this bacteria, pokes holes, lysed. This is a view of the classical and alternative pathway of the complement system. We mentioned the classical pathway. Basically, these antibodies have recognized this as foreign, as a threat. So this summons C1, which eventually activates C3 into C3b. This activated C3b has all of these functions. It attracts phagocytes, stimulates phagocytosis, stimulates inflammation, as well as summoning all of these other components, C5 through 9, to create these pores, this membrane attack complex, these MACs. So that this is the classical pathway, antibody mediated. You also have an alternative pathway that is activated through properdin, factor B for properdin, factor BD, that also stimulates C3, activates C3 into C3b. And once you activate C3b, you activate C5 through 9, that allows the formation of the membrane attack complex, pokes holes on the bacteria, foreign invader, and eventually lyses it. Okay, so far we're looking at non-specific lines of defense. Whenever we let's talk about inflammation as one of these non-specific response. This is what happens with inflammation, redness, heat, pain. These the end results of it is going to be whenever you summon components of the immune system, have repair of the injury. This is done through platelets, not components of the immune system. You're going to slow the spread of pathogen, and you're going to this is part of the immune system that's going to be summoned, and it's going to hopefully stop this. The inflammatory response mediated by mast cells releasing interleukin, cytokines, and so forth. This is the end result, is going to increase blood flow, activate phagocytes, increase capillary permeability. This allows white blood cells to migrate from the lumen of blood vessels into the site of injury. You also have fever as a non-specific mechanism. Pyrogens basically stimulate your thermostat within your hypothalamus, and this allows the temperature to be raised through your just an increased metabolism. Just like whenever you run, a lot of ATP is being consumed. ATP just like whenever you run a motor, there's no just perfect transduction and perfect translation from these high energy molecules into work. Some of it is lost in heat. We lose it in heat. Uh, the we use a similar mechanism of consumption of ATP in order to create that heat, increase our heat in our bodies. And that heat, usually our cells, we have these chaperone proteins that allow us to be somewhat heat resistant. The chaperone proteins mm, allow our proteins to still function. Uh, Apparently, viruses and bacteria are not as fairly they're not as resistant as we are. So let's talk about this. Okay, specific defenses. Another break, take a break. I encourage. One more, take a nap if you want. Whenever it comes to learning, naps are uh, are a, a are a great companion for learning. Basically, whenever you sleep, whenever you nap, your brain is consolidating some of this knowledge that you have acquired. So, catnap. Um, okay, let's look at the specific this specific immunity. We're going to look at different types of immunity. These are straightforward. You're going go through this and you will understand them. They're fairly self-explanatory. Let's look at properties of immunity. It has to be specific. That's in order for it not to attack us. It has to be versatile in order for us to just, through randomness, create a lock and key mechanism with foreign invaders that we have never been exposed to. It has to have memory. That way, whenever we get exposed to it again, boom, we're gonna get it. And tolerance. Tolerance so it doesn't attack us. So let's look at the immune response. Antigens trigger the immune response. You're going to hear me talk about antigens and epitopes almost synonymously. There's some technical distinctions between them. Epitope, I think, is more specific. But let's, for the sake of now, let's just go with that, with the synonym aspect of it. Phagocytes. So basically, this is what happens. You have an antigen, foreign substance. You have a phagocyte, eats it. It says, "Hey, I ate this. It doesn't taste good." It puts it on the surface so that a lymphocyte, which is the cavalry of the immune response, can help us like truly attack it. So you have your T cells and your B cells that are being summoned. The T cells gets activated. T cells attack the antigens. And remember the helper T cells? The ones that say, help, help. They stimulate the B cells. And this is a check and balance step of the immune system. If the B cell has also recognizes this, and there is this agreement, like I've seen this is not a good protein, this is not a good antigen, let's attack it. So in this case, the B cells will mature into plasma cells, and they're the ones that secrete antibody. Once we get to the antibody portion, we kind of it's analogous to going from like hand-to-hand combat to get the archery, and they can attack with many arrows, aka antibodies. Okay, so this is a nice picture of it. Antigens, here they are. This is most likely, is that a coronavirus? I'm not sure. It looks like it has a nuclear. All right, beyond the point. Here you go, specific defenses. You have your phagocytes doing this digestion, presentation to T cells. T cells with helper T cells telling the B cells, activating the B cells. uh, match, differentiating, maturing into plasma cells, and they releasing these antibodies that attack the T cells. T cells themselves, through cytotoxic T cells, are going to attack the foreign invader. Excuse me. Excuse me. Okay, it's late. I'm a little delusional at the moment. Okay, let's go through. Let's talk specifically about the activation of T cells through these antigen presenting cells. Okay. There's a lot of this process is highly specific, and there's a lock and key mechanism occurring between the antigen presenting cell, and the T cell itself. So all of those three components must recognize this particular antigen. These two cells must recognize this particular antigen. Let's see how that works. Okay. So first, the major types of T cells. I think we talked about them earlier, but let's refresh. Cytotoxic T cells, the ones that do the attack. Your helper T cells, the ones that activate T cells and B cells. AIDS patients have a reduced numbers of T cells, so they basically cannot create this activation, this like summoning of large portions of the immune system of B cells and T cells. Memory T cells are the ones that keep basically they keep memory of this antigen. So this antigen ever shows up again, we'll just, hey, I have that cell that knows how to that creates an antibody, so we clone it, and we have a faster response. And suppressor T cells that moderate the immune response. Okay, let's talk about antigen presentation. So a macrophage, an antigen presenting cell, a dendritic cell, microglia has eaten something that didn't taste good. Recognize the antigen, and they place it in a glycoprotein. A glycoprotein called a major histocompatibility complex protein. So this foreign antigen gets placed on this protein and gets presented. There are two types of MHCs, of major histocompatibility complex proteins. The class 1 is found on all cells, and it basically, whenever a cell presents that in its surface, it says it has a signal that says, kill me, I'm infected by this virus, please kill me. Or if you have class 2 MHC cell, this is found on antigen presenting cells. So basically they're presenting this antigen and saying, kill this. So class 1, I want you to just memorize that right now. Take your time to memorize that, because otherwise things are gonna get confusing. So basically, antigen presenting cells that have eaten cells of the immune system, cells that can have this phagocytic activity, they have digested an antigen, a foreign substance, and they are placing it on these major histocompatibility complex proteins on its surface, saying, look, I found this, kill it if you see it. Now, these are, cells of the immune system with phagocytic activity. Now, non-immune cells, all of the other cells in your body, whenever they get attacked, they also present within their surface this particular foreign substance, and they say something's wrong with me, basically. Or on their surface, they usually, in this MHC1 protein, they usually in this in this surface protein, they usually present uh, substances that say, "Hey, it's me. I'm all right. Don't kill me." But whenever things go awry, then usually that changes, and this cell is no longer recognized as our own, so it gets attacked, it gets killed. Okay, so I hope that you understand that distinction. If so, let's class one is a way that cells that get sick tell other cells of the tell cells of the immune system to kill them. So they say, "I've been infected by a virus. Please kill me." Um, and cells class two major histocompatibility complex, they digest a virus and they say, "Kill this." All right. So let's in this example, we have a cell being attacked by a virus. The cell digests the virus, and then it presents this on its surface. This is a signal saying that there is something wrong with this cell. So a viral attack can happen to any cell of your body. So any cell of your body can do this, send a signal that says, "I'm being attacked by a virus, please kill me." So in the case of COVID-19, you have cells binding to the angiotensin receptor within your lung cells. I think it's the pneumocytes. In binding to this receptor, once they bind, they inject their viral, this particular retrovirus. It divides. The cell itself is probably um, is probably a recognized kind of digesting some of these viral components and presenting them in its surface. Like, this is COVID-19. It's inside of me, and it's presenting it through these major histocompatibility 1 proteins. And this, what they're saying through this major histocompatibility 1 glycoprotein, it's saying, "Please kill me. COVID-19 got to me." On the other hand, if a particular if an antigen presenting cell happens to come across COVID-19, it digests it, and then it breaks it down, it recognizes it as a foreign substance, it presents it on its surface through major histocompatibility complex proteins, glycoproteins class 2, it says, "Hey, I found this, we need to kill this." So these phagocytic cells, these antigen-presenting cells, they do whatever the name suggests. They present the antigen. Who do they present the antigen? To the to the to the lymphatic system. Who is the workhorse of the lymphatic system? Lymphocytes, B and T lymphocytes. All right. So now we have identified how these antigens get presented, how cells say, "Hey, I'm being infected by a virus, natural killer cells, please do your function and kill me." Or they say, "Hey, I found this particular virus, it looks like this, it looks like COVID-19, please find it." It shows it, it presents it to lymphocytes and says, "Please find it, please kill it." Now let's look at how the lymphocytes recognize this, because it's a lock and key mechanism. that allows this interplay to occur. So let's look at the specific components that allow this recognition. So here we have the lymphocytes that are at the other end, your natural killer cells, your B lymphocytes, your T lymphocytes that are at the other end, waiting for that signal in order to to perform their immune function. They do so by these membrane components called clusters of differentiation. Okay. These clusters of differentiation we have two kinds. We have three kinds, but I want you to focus on these two. This third one will speak about it in a little bit, but these are the ones that I want you to focus on, because these are the ones that are going to interact with the major histocompatibility complex 1 and major histocompatibility complex 2. So we talk about the major histocompatibility complex 1, the one that says kill me. Who does the killing? Natural killer cells do the killing, right? So it will present it to so the cells that will have the lock and key mechanisms would be natural killer cells. On the other hand, whenever you have natural killer cytotoxic cells, basically cells that can kill something. They're the ones that are going to be recognizing it. Basically, cells that can kill a cell. They're the ones that are going to answer that plea by infected cells, that by virally infected, by cancerous cells, that says, please kill me. Okay. So here you have, on the other hand, you have cells that present through major histocompatibility, antigen presenting cells that present through major histocompatibility complex type 2 and antigens, and it's saying, please kill this. So who does this particular function? It's going to be B cells and T cells, but they won't do it until the helper T cells tell them to do it. The helper T cells are the ones say, hey, help, we found this, help, help me T cells, help me B cells to kill this. All right. So one of the things, if you want to know like which major histocompatibility complex binds to which CD complex, cluster of differentiation complex, if you multiply them, they all come to 8. CD8 binds to MCH1, that's 1 times 8, 8. CD4 binds to major histocompatibility complex 2, that's 2 times 4, that's 8. Remember that on your exams. Okay. So let's look at the activation of the CD8. So basically the CD8 is on cytotoxic cells, on cells that recognize cells that are pleading to be killed. Okay. So this is what's happening. Infected cell begging to be killed. So here you have lymphocyte that has this cluster of differentiation 8. You notice that the cluster of differentiation is over here. This T cell receptor, that is the MCH class 3 that I didn't want to talk about it. Just so we don't get confused, let's call it T cell receptor. But just know that it is that other major histocompatibility complex. We'll call it T cell receptor. Okay. The T cell receptor, through this lock and key mechanism, recognizes this antigen, and there is this CD8 kind of creating this kind of check and balance, that it would only recognize this class 1 major histocompatibility complex protein. glycoprotein. Okay, so anyway, finally we have a cell that's beginning to be killed through its major histocompatibility complex, um, presenting the antigen that is culpable of this. And we have the T cell, cytotoxic T cell, finally binding and ready now to perform this killing. Now, how does that happen? You have your cytotoxic T cells releasing cytokines that stimulate apoptosis of or programmed cell death. What's the difference between programmed cell death and necrosis? Necrosis is not programmed. If necrosis, basically the cell just loses all homeostatic uh, balance, and like its contents get spilled into the exterior. That's not beneficial to the cells that remain alive around it. Um, it's like performing a demolition of the house right next to you. Whereas apoptosis is basically the cell, it starts it starts in an organized manner, um, in creating these digesting all of its interior components, creating these vessels, that the vesicles that eventually get recycled by macrophages. So everything neatly dies. So that can be induced by this cytokine release of the virally infected cell. We can tell the cell, hey, uh, go ahead and we stimulated the cell death. We allowed the cell to die in a good way. You also have release of lymphotoxin that disrupts the cell metabolism. Usually, we don't want these cells to continue metabolizing either viruses or cancerous component. We also have destruction of cell membranes. All of these things are happening simultaneously. It's kind of like if this fails, this will happen, this will happen. Okay? So throughout this process, you have your cytotoxic T cells being activated, but you also have your memory cells being your memory cells being formed. So this once this lock and key mechanism happens, you have division of these T cells, because usually it's not just one cell that gets infected by a virus. So the cavalry arrives, attacks all of the cells that are infected by a virus. They either lyse it, or they allow it to die a good death through apoptosis, and then they create these memory cells that remember that antigen for next time they see it. Okay, so this is what happens whenever cells get infected by viruses and request to be killed. Now, let's look at what happens when an antigen presenting cells identifies a foreign substance. So, if this particular antigen presenting cell has digested a foreign substance and it's placed on the surface of its major histocompatibility complex class 2 protein. Remember, 2 times 4 equals 8. That's why class 2 binds to CD4. Okay. So here you have the T cell receptor in the lymphocyte. So this is the antigen presenting cell presenting to the helper T cell, and through this T cell receptor, it recognizes it. This says, and this CD4 protein also plays a role within this recognition. So basically, we have come to an agreement. This is a foreign substance, and this is the T cell that recognizes it. Basically, not every T cell, not every helper T cell will recognize this antigen. Only specific T cells would recognize this antigen. That's why we're creating memory cells, right? It's because this is a very, very specific type of recognition of the antigen. And that's why we create those memory cells, so that next time the antigen presents itself, those memory cells have have this lock and key mechanism to recognize this. This all happens through just genetic variability and randomness. We have like trillions of these particular cells um, form, and at any time there's like 100 million of these cells like creating that are able to recognize 100 million different foreign substances just through randomness. So here you have finally the antigen present cell to the helper T cell. It recognizes it. Now the helper T cell, what does it do? Help, help. It requests help from. It activates basically B cells and T cells themselves that to not only create memory cells, but to create cells that we're going to be able to create the antibodies or attack the cells that are affected by this. So active THC cells, they will release cytokines and memory THC, memory helper cells. So this is what helper T cells do. They release lymphokines, enhance they do all of this. So. All right, this is a summary of what whenever these recognitions occur, what happens. These are cells that are asking to be killed. This is antigen presenting cells requesting that they kill this antigen that they have presented. So these helper T cells stimulate the response of T and B cells. They say, help, help. And then they create memory cells that if this happens again, they will be able to respond faster. Whenever it comes to cells that request to be killed, you have whenever you have this recognition occurs, you create the memory cells, you create the immune response that attack these particular cells or antigen, and then they attack the particular cells, and then you also have suppressor T cells that don't maintain range on the immune system. All right, so that was the T cells. Now we're gonna look at B cells. These are the ones that are able to create antibody. These are the archers of the immune system. So let's get started. So you have this process of sensitization occurring. This process of sensitization allows antigens to be kind of presented in the surface of these particular B cells. You also this will allow the interaction with basically antigen presenting cells. You have T cells themselves, helper T cells activating the B cell through cytokines, and this is a process of co-stimulation. So basically, the B cell, the antibodies in the surface of B cells recognize a particular foreign substance. And you have a T cell a helper T cell that has found the same substance. Basically, the T cell interacts with the B cell. The helper T cell interacts with the B cell, saying, "Hey, I found this. I see that you saw it too. We are in agreement. This thing needs to go." So that process, this co-stimulation is kind of like a check and balance that allow the immune system to allow. There's like a bottleneck at the helper T cell level. That bottleneck that explodes into a giant activation of B and T cells. But this activation has this co-stimulation, this like check and balance to it. So let's look at this process. So um, you have here this antigens kind of binding. You have these B cells that create a particular kind of antibody, a specific type of antibody. They only create one type of antibody, each T cell, each B cell, I'm sorry. And whenever that antibody gets stimulated by an antigen, you have you have a similar process of presentation of this antigen in through through your major histocompatibility complex. So in this case, major histocompatibility complex type 2. So in this case, the B cell is kind of serving that role of an antigen presenting cell to the T cell that contains that cluster of differentiation form. So there is agreement that between the helper T cell and the B cell, and this leads to the formation of memory cells and also the rapid cloning and expansion of this particular uh, B cell that we know recognizes this antigen because that's how it was stimulated to begin with. And this B cell differentiates into plasma cells. And at the plasma cell level, that's whenever antibody production just goes sky high, and that the titer of antibody continues growing to the level that we can fight the infection. Let's look at the structure of the antibody. We are almost there. We're like we're 17 slides away, but they're gonna go fast. Okay. So here is let's look at the structure of an antibody. You have a constant region and you have a variable region. The constant region is what's going to determine what type of antibody is. We're going to look at the different types of antibody. Your variable region, that's due to the just genetic splicing, different splicing of these particular genes, just randomly recognizes something else. It's just like different amino acids give it different electrochemical characteristics that allow it to recognize epitopes, antigens from bacteria, antigens from viruses that it's never seen before. Look at the antibody. It doesn't seem to have like a complex structure to be able to just create custom regions that would recognize whatever antigen it wants. The way that the immune system works is just like creates a lot of B cells, and one of them is going to create the antibody that's going to recognize the foreign substance that's attacking us. So it's kind of like a quantity. It's all about quantity in this case that allows these antibodies to recognize foreign substance. So here you have two chains. You have your heavy chain and you have your light chain, but you also have within this, if you cut them in a different way, you have your constant region and you have your light region. Your constant region and you have your variable region. I'm sorry. The variable region is the one that gives the antibody its specificity. a constant region is what gives the antibody its different type. Here you have, anyway, this is the antibody side in the variable segment. So once an antibody recognizes a foreign substance, what are its functions? It neutralizes the particular substance. For example, if it neutralizes the crown region of the coronavirus, then the coronavirus can no longer bind to its target. So imagine if it's a lock and key mechanism, if the key from the coronavirus, you put something analogous to bubble gum on its surface, then it won't be able to properly align with the lock. So uh, the gum analogy kind of works because these proteins, they're kind of sticky. So that's why agglutination or these precipitation reactions kind of happen. They just like stick everything together. They have this particular property. They can activate the complement. We talk about the classical pathway of the complement activation that allows the complement to poke holes within the surface of cells. It attracts phagocytes. It leads to opsonization, which is it promotes phagocytosis. It promotes digestion. It promotes stimulation, and it prevents addition of this by the blockage of the epitope. It prevents addition. This is an example of a hapten. It's just an immunologic. It's a concept about usually. So you have two variable regions within this particular antibody, and usually those two variable regions will recognize the foreign antigen. A hapten is whenever only one of the antigen is just recognized by one. So sometimes a carrier molecule allows the complete recognition of it, and it could lead to some sort of improper immune response. But anyway, don't worry about haptens right now. Classes of antibodies. We have 5 different classes of antibodies. IgG. That's the one that's mainly responsible for the bulk of your antibody humoral response. You have IgE found on the surface of basophil and mast cells. They're the ones when something binds to them, they release all of these vesicles containing histamine and this type of interleukins that promote the inflammatory response. They're responsible IgE through basophils and mast cells through vesicles filled with histamine are responsible for your allergies. IgD is found on the surface of B cells. Remember how we talked about how B cells get activated? It's through the IgDs on their surface that recognize foreign antigens, that recognize specific antigen. You have IgM. This is the first responder of the immune system. And it's responsible for blood agglutination. This is the one that recognizes the different the blood antigens. And you have IgA found in granular secretions, basically released into mucous surfaces. This is the primary and secondary antibody response. Your primary response, the first time you have been exposed to the particular antigen, you have an increase of IgM, a subsequent larger increase in IgG. Now, if you get exposed to this particular antigen once again and you have memory cells, these memory cells are going to immediately undergo this clonic expansion. They're going to divide, clone themselves over and over again, and immediately create this antibody response. So it's going to be faster, more sustained. Oh, there it is again. Let's ignore it. In summary, an antigen arrives. You have your non-specific defenses. You have your fevers, your interferon, you have your complement, you have your natural killers, your macrophages. Some of these, you also have your specific response. That is the activation of T cells or B cells through helper cells, and you have these suppressor T cells moderating this response. Activation of B cells are going to lead to the production of memory cells and B cells or plasma cells that produce antibodies, and the attack of antibody attacks the antigen. And T cells, production memory cells, and T cells that produce these toxic, cytotoxic T cells that undergo direct contact with the antigen. So this is whenever we have the response towards bacteria involve the activation of a macrophage digesting the bacteria, then presenting the antigen, activates the cytotoxic T cells that go and fight the bacteria one-on-one, activates the helper T cells in case the B cells have found the same antigen, and then that leads to the production of antibodies and a sustained response. In the case of viruses, they infect the tissue. This particular cell can say, hey, I'm sick, so there can be a response by interferon. There can be antibodies, the antigen of the bacteria presented on the surface, stimulating natural killer cells or cytotoxic T cells. So there's also the virus could directly be phagocytized by viruses. Then there is the antigen presenting cells stimulating helper T cells and B cells and so forth. All right. Don't know that there are interleukins and know that there are interferons that are antiviral for tumor necrosis factors, slow tumor growth, colony stimulating factors. They stimulate lymphoid stem cells. So know this main function. You probably will hear of this repeatedly. You probably have heard already of interferons, tumor necrosis factors, interleukins. So this is a good time to just kind of digest them so that whenever you get presented with them in the future, you'll have a place to store that. Immune disorders, allergies. Whenever it comes to allergies, let's just go through this. That's what we need to know about allergies. So there is the first exposure to this particular allergen. Macrophage gets exposed to it, and it undergoes kind of like a similar process as any antigen. It activates helper T cells, sensitized B cells, plasma cells that create antibodies. Now, these antibodies, these antibodies created by plasma cells, the IG antibodies are going to localize themselves to this either mast cells or basophils that contain all of these inflammatory vesicles here containing histamine. The next time that there is an exposure, this antibody is going to bind to this particular antigen, and it's going to lead to the release of all of these histamine and other inflammatory mediators, and that's what you're gonna get. Major sniffles over the spring and summer. Stress is gonna mess with our immune system. It activates this stress. It's going to the, having a prolonged stress response is going to affect our immune system. Let's just leave it at that. These are some of the of the what could happen: depression, the inflammatory response, phagocytic reduction, inhibition of interleukin. Aging, just like stress, aging, if you age, your immune system is going to decrease its function. All right. I hope you made it through this in several sittings. I don't know how to make this shorter without me just breezing through it. But anyway, have another listen to it because it's a lot of material. It's let's go through it together once again, and you also rely on your book. There's a lot of detail within the book that will help you cement the generalities of a lot of this process. All right. Thank you for your attention and hope you have a good rest of your day.